Hello everyone, my name is Dimitri Perrin. It is my pleasure to talk to you about work we've done as part of the CAMDA 23 challenge. This is work completed with my colleagues Shlomo, Colin, Steve, Tim and Jim at QUT and CSIRO. The backdrop for the challenge is antimicrobial resistance. AMR is a very pressing health issue. It is estimated that more than 1 million people die every year from antibiotic-resistant bacterial infections. It is a growing concern globally, but it's also a very diverse one, including in terms of which species are going to be problematic in different regions. If we look at this recent paper, we can see that different species will have a different prevalence in different countries, but even within the same species, the patterns of antimicrobial resistance and which antibiotic the infection is going to be resistant to is going to be different and change quite widely in different regions of the world. And so we can ask ourselves how far that geographical dimension goes. The idea of the CAMDA challenge is to take some hospital data from a single location and try to see if it is possible, just based on AMR patterns, to understand and predict where that hospital is located. Let's start by looking at the data that was available through the challenge. On one hand, we have whole genome metagenomics datasets available from a number of cities. They came from the Metasub consortiums, and here we're going to focus on locations in the US. So we have a number of cities in the US, and for these cities, we have any number of samples that were collected in different locations in these cities. On the other hand, we have AMR markers from close to 150 isolates from a single hospital that is located in one of these American cities. And the task we set for ourselves was to take data from the first group and see if we can use that to predict the location for the hospital. For the AMR markers, the data comes in the shape of an Excel spreadsheet, where for each of these 146 isolates, we have the species and we have some AMR markers. What we need for our approach is the underlying sequence for these markers. So we went to the CARD database and extracted that information. Of course, most of these markers are not specific to a species, but here in the way we construct our reference collections, we only use markers for which in the CARD database, we're able to find the same marker on the same species as the isolates from the hospitals. In total, this gave us 78 reference sequences. So now we have a reference collection that contains 78 CARD reference sequences. What we want to do is query that reference collection using reads from the Metasub samples. And for each of these reads, we want to identify whether it is a match to that reference collection or not. Of course, because of these Metasub samples contain whole genome sequencing, we expect that most of them will not be matches, but we want to identify the ones that are matches to the collection. That Metasub data is quite large, so our method is focused on going through that data as quickly as possible. Part of the solution is to use signatures. For any given sequence, we're going to extract k-mers and project them to a smaller dimensional space. Here, we project onto 24-bit integers, and that's going to be sufficient because we have a small number of unique k-mers in the CARD reference collection. If needed for very large collections, we could go to 32-bit or 64-bit signatures. It wouldn't fundamentally change the approach, and that would be sufficient because even for large collections, the set of all possible k-mers in that collection is still going to be much smaller than 2 to the power of 64. The key advantage of using signatures is that it's going to give us flexibility on the k-mer length. We can use the exact same architecture for our methods for very short k-mers all the way to very long k-mers, or even using the whole read as an input. These signatures form the backbone of our approach, called k-mer indexed signature search, or KISS. We start by selecting k-mers from the read and looking them up against the index built on the CARD reference collection. If after several attempts we cannot find a seed in the index, we discard that read from further consideration. It is not going to be a match to the collection. If we can find a seed, then a local alignment is performed to attempt a match. If a suitable alignment is obtained, the read is admitted as a match to the collection. If not, we try to find a new seed and repeat that process. Of course, at a high level, this is not completely different from other existing approaches, but our contribution is going to be in how we can accelerate all of these steps. One solution could be to slide a window one base at a time through the read, and for each of these windows, try to find the corresponding seed into the index. It would work, but we can be faster, both in terms of how we move that window, but also how we attempt each seed lookup. In the seed lookup, we want to try to see if that k-mer that we extract from the read is present in the reference collection. In our case, the collection of relevant card AMR sequences. We can make this faster by minimizing the number of times we calculate the signature. To do that, we use an indicator matrix. In the matrix, each bit corresponds to one k-mer in the reference collection and indicates whether that k-mer exists in the collection or not. The position of the indicator bit for a given k-mer is calculating by splitting it in half, converting each half into an integer, and using these two values as the row and column index of that k-mer in the indicator matrix. That conversion of half the k-mer into the integer is just a straightforward radix transformation, so it's quite efficient. To illustrate how this works, let's imagine that we work with dimers and that we have this reference sequence. Of course, half a dimer is going to be a monomer, and we have this simple radix transformation, A to 0, C to 1, G to 2, and T to 3. This transformation is going to give us the index positions into the matrix. We can use that to fill the bits in the matrix based on our reference sequence. For instance, AC is not present in the sequence, so we get a 0 at position 0, 1. GA is present in the sequence, so we get a 1 at position 2, 0. And similarly, TG is present in the sequence, and so we also get a 1 at position 3, 2. Of course, in practice, we don't work with dimers. If we use 16-mers for the indicator matrix, we split each of them in halves, so we get 2 8-mers. Each half is then transformed on the range from 1 to 4 to the power 8, and that gives us 2 index position into a sparse matrix. We could do the same thing using an indicator array as well, instead of a matrix, but here we know that because of the collection that we're working with, most of the 16-mers will not be present, and so there's an advantage to using a sparse matrix implementation. The next optimization is in how we skip through the read to identify seeds. For the purpose of the discussion, let's imagine that we're looking for a match of length 64. In practice, the length of the match is something we can change easily as a parameter of the method, but using 64 is convenient for the discussion here. So we start at the beginning of the read, and we want to identify seeds that we can use to then perform an alignment to find a match of length 64. So we're going to split that window of 64 into 4 fragments of length 16. We can quickly check for the presence of these fragments into the reference using the indicator matrix. In particular, we're considering D as a potential seed. If D is not present in the reference collection, then we know for sure that no window that contains D can be an exact match to the reference. We use that to skip completely past D for the next attempt at finding a seed. So if we start here at the beginning of the read with that fragment D as a potential seed, the next window we consider is the one that starts at one position after the start of that fragment. It's important to note that D not being present in the collection is the most frequent behavior in our use case. Remember that the reference collection only contains AMR sequences, whereas the queries are reads from a whole genome metagenomic sample. So we expect that most reads will not be a match to the collection, and that gives us a way to quickly go through these reads that are not going to be in the collection at all. If D was found, we use that as a seed and attempt a local alignment. If that local alignment is successful, we accept the read as a match and move on. That local alignment can be successful even if we have mismatches, but for sure, if it is unsuccessful, then it is because we have mismatches in A, B, or C. And we're going to use that information to skip the window again. We're going to skip to the position just after these mismatches before we look for a seed again. Our first attempt was this window at the start of the read. We identified that D was present in the collection, performed the local match, and it wasn't good enough. We're going to move through the read to a new position and start looking for another seed. And we move past some of these mismatches because we know that we had enough mismatches that the local alignment wasn't good enough. And so we want to move far enough into the read before we attempt new matchings. By combining these two skipping strategies for when D is present or not, we can process each read very quickly. When we identify a seed that is present in the collection, the local alignment can be a very flexible step. You can perform it using any suitable method, for instance, Smith-Waterman or straight up string comparisons or any other alignment method. The key thing here is that we keep the cost of that alignment very low for two reasons. Number one, we only consider that local alignment if we identify a seed. And for most reads, we will not have a seed because that read is not an AMR sequence. Second, when we do have a seed, then we know which region of the collection is a potential match, and the alignment is restricted to a very small region. As I mentioned earlier, if the alignment is good enough, we consider that the read is a match to the reference. And one good alignment is sufficient to consider the read a match. We do not do an exhaustive search. So overall, we have the following strategy. For any position in the read that we are currently considering, we start by looking for the seed using the indicator matrix. If the seed is not present, we skip along the read. If it is present, we perform a local alignment. If that alignment is good enough, we accept the read. If it is not, we skip along. After several attempts at finding a seed and performing local alignments, the read is either going to be accepted or rejected. Before looking at the result, there is one more aspect of the data that we need to discuss. For each city that we consider, there is a variable number of FASTQ files. And the size of these files also varies greatly from just a few million reads in the smallest files to over 50 million reads in the larger ones. For instance, New York City has almost 70% of the total number of reads in the collection that we're working with. Of course, the more reads we have, the more AMR reads we're going to identify as well. And so we need to normalize our results. The way we do this is by scaling the number of matches that we identify for a given city by the ratio of the number of reads from New York compared to that city. Choosing scaling instead of rarefaction avoids data loss, and it's possible given the amount of data that we have. Part of our future work is to consider other normalization strategies and evaluate the robustness of the approach. Using our method, we analyzed every read from every sample from all of the American cities that were available, and we report some results here. What we're showing on this plot is the scaled number of matches for a given city for the 78 card reference sequences. There are two main things worth highlighting in this plot. First, New York has the highest number of matches overall. And second, Baltimore has the highest number of sequences for which we can find a large number of matches. Next, we looked at how we perform the local alignment after the identification of the seed, and in particular, whether to allow mismatches or not. By default, we do allow mismatches. That's the plot I showed previously, and that's the one reproduced on the left again. But we can also force exact matches. That's the plot on the right. As we just discussed, in the default setting, New York has the highest number of matches, and Baltimore has the highest number of sequences with a large number of matches. If we force exact matches, Baltimore still has the highest number of sequences with a large number of matches, but it also has the highest number of matches overall. Based on these two sets of results, we made the initial prediction that the hospital was located in New York or Baltimore, and the challenge organizer just announced that the hospital was in fact located in Baltimore. Of course, at the time when we were developing the method, we did not know what the hospital was. So as a sanity check, we wanted to compare our results to an existing method. What we did for comparison is to take reads from the metasub data and try to align them to the guard sequences using BotI2. As you can see, qualitatively, the results are very similar. The main difference, though, is the time that it takes to produce these results. We performed our test on a computer that has 48 processors and 96 cores. For BotI2, we found that the best way to fully exploit these resources was to run with 128 threads. If we do that, it takes just under 4.5 hours to assess the whole collection. Our method was implemented as a simple MATLAB prototype, and because of that, it cannot fully exploit the 96 cores it's running on just the 48 processors. Despite that, it runs in less than 2.5 hours on the same collection. Since then, we've started working on the C++ implementation, and early results are already much faster than this. In summary, we started with a simple question. Can we use AMR patterns to distinguish between cities? Are these patterns localized enough to make this possible? To do this, we developed a method that can rapidly process very large sets of whole genome sequencing data and identify matches to a reference collection. We showed that for the available data, we were able to make a reasonable prediction. The initial prototype for our method was already fast, and we are working on making it much faster. And of course, even though the method was developed as part of this challenge, it has applications that go well beyond this context. As an example, many datasets need to be filtered to remove possible sources of contamination. For instance, trying to remove human reads from a bacterial dataset. With our method, we can do that very quickly. And with that, the only thing left for me is to thank you for your attention. If you have any questions, please do not hesitate and get in touch.